Once upon a time, in a world not too far away, there lived a brilliant scientist named Dr. Smith. Dr. Smith had always been fascinated by the mysteries of life and had spent years studying genetics and the secrets hidden within our DNA. One day, while working in his laboratory, he stumbled upon a groundbreaking discovery that would change the course of science forever. He had discovered a way to clone living organisms. Cloning, in simple terms, is the process of creating an exact genetic copy of an individual or organism. Dr. Smith's discovery opened up a world of possibilities, from potentially finding cures for diseases to preserving endangered species. Excited by the potential of his breakthrough, he began working tirelessly to refine his cloning technique. Step 1. Gathering genetic material. The first step in the cloning process is to collect genetic material from the organism that will be cloned. In Dr. Smith's case, he chose a mouse as his subject. He carefully extracted a small sample of cells from the mouse's body, typically taken from its skin or muscle tissue. These cells contain the complete genetic blueprint of the organism, stored within its DNA. Interesting fact: did you know that the concept of cloning dates back to ancient times? The ancient Greeks and Egyptians had myths and stories about the creation of living beings through asexual reproduction. Step 2. Isolating the DNA. Once Dr. Smith had obtained the cell sample, he needed to isolate the DNA. DNA, or deoxyribonucleic acid, is a complex molecule that contains all the genetic information needed for the development and functioning of a living organism. By carefully extracting the DNA from the cells, Dr. Smith could access the instructions that would allow him to recreate the mouse. Step 3. Inserting the DNA into an empty egg. With the DNA in hand, Dr. Smith obtained an empty egg cell from a female mouse. He carefully removed the nucleus from the egg, which contains the egg's own genetic material. This created a space for the introduced DNA to take its place. Using specialized techniques, he inserted the mouse's DNA into the egg, effectively replacing the egg's original genetic instructions with those of the cloned organism. Interesting fact: the first successful cloning of a mammal, Dolly the sheep, took place in 1996. Dolly was born on July 5, 1996, at the Roslin Institute in Scotland. Step 4. Stimulating cell division. Once the DNA was successfully inserted into the egg, Dr. Smith stimulated the cell to start dividing. This was done by creating the right conditions for the egg to grow and develop, just like a naturally fertilized egg would. The egg was placed in a nutrient-rich environment and provided with the necessary conditions to encourage cell division. Step 5. Implantation and development. After a few days, the cloned embryo was ready to be implanted into a surrogate mother. Dr. Smith carefully transferred the developing embryo into the womb of a female mouse that was capable of carrying the pregnancy. The surrogate mother would provide the necessary environment for the cloned embryo to grow and develop into a fully formed organism. Interesting fact: cloning doesn't always result in an identical copy of the original organism. Environmental factors, such as diet and lifestyle, can influence gene expression, leading to slight variations in appearance and behavior. Step 6. Birth of the clone. After a gestation period, just like any other pregnancy, the surrogate mother gave birth to a healthy, cloned mouse. This mouse was an exact genetic replica of the original mouse from which the DNA was obtained. Dr. Smith had successfully cloned a living organism, a feat that had never been achieved before. The news of Dr. Smith's groundbreaking discovery spread like wildfire throughout the scientific community and the world. Cloning became a topic of intense debate and raised various ethical questions. Some saw cloning as a way to revolutionize medicine and find cures for debilitating diseases. Others worried about the potential for misuse and the ethical implications of playing with the very fabric of life. Interesting fact: cloning has been successfully performed on a variety of organisms, including plants, animals, and even human cells. However, human cloning for reproductive purposes remains highly controversial and is generally considered unethical. In the years that followed, cloning technology continued to advance, and scientists cloned various species, including dogs, cats, and horses. However, many challenges and limitations still exist, and cloning remains a complex and specialized field of study. Dr. Smith's journey into the world of cloning was just the beginning of a scientific revolution. While the story of cloning is not without its controversies and ethical dilemmas, it continues to hold promise for the future of medicine, conservation, and our understanding of life itself.